today's video we will look at a very interesting concept called synthetic lethals. So, synthetic lethals are sets of genes or reactions where you need to remove all of them together to abolish growth in a cell. So, if you remove a single gene nothing happens, but if you remove a pair of genes the organism dies. So, how do you identify these combinations from a metabolic network? We will go into greater details in a moment. Okay. Welcome back. We have been looking at perturbations to metabolic networks. Let us look at an important type of perturbation. We have looked at deletions and overexpressions. Let us look at another important type of perturbation which is multiple deletions, and which leads us to the concept of what is known as a synthetic lethal. So, what are synthetic lethals? So, they are sets of reactions or genes when only the simultaneous removal of all the genes in a set abolishes growth of the organism. So, let us consider the simple example here. So, there are two genes ABC and PQR. When you delete ABC, as you see in the bottom, nothing happens. When you delete PQR alone, the organism still grows. When you delete both genes together, the organism dies. So, you synthetically superimpose the two deletions essentially, right, and you can therefore you can also extend the concept to higher orders, right. So, let us say there are reactions R1, R2, R3. When you remove these two nothing happens, when you remove these two nothing happens, when you remove R1 and R3 the organism continues to survive. When you remove R1, R2, R3 it is lethal, right. So, any you can also therefore extend it to quadruplets as well. If you remove 3 at a time, 2 at a time, 1 at a time nothing happens. When you remove 4 at a time the organism no longer survives, right. This concept is known as synthetic lethality. So, why do we need to identify synthetic lethals? What are the use cases? First thing, it helps us understand gene function and functional associations, right. So, it it is kind of easy to find out what genes are isozymes for one another and so on, but through this analysis you can find two reactions that are in different subsystems that could potentially be compensating for one another, right. So, it is possible to find genes or reactions that are sort of far apart which can compensate for one another. Synthetic lethals are also useful to identify combinatorial drug targets and pathogens, right. So, this is again a matter of intense research and debate. So, it is common to have organisms especially bacterial organisms develop resistance, right. You must have heard of especially in diseases such as tuberculosis, you must be familiar with many of these acronyms. There is MDR, XDR, TDR, multi-drug resistant tuberculosis, extensively drug resistant tuberculosis, totally drug resistant tuberculosis, resistant to all known drugs. Similarly, you have MRSA, VRSA and so on, methicillin resistant staph aureus, vancomycin resistant staph aureus and so on. Right. The idea is, could you in any of these cases can you target more than one gene, right. So, this makes it that much more difficult for the organism to develop resistance, but that still remains a question, should you target a synthetic lethal or two single lethal genes, right. You remove A, say you remove G1 the organism dies, you remove G2 the organism dies. You could also look at this as a pair to target, right. But the problem is there is a lot of benefit to adapt to this mutation, right. So, in other words, whenever you throw a drug at an organism you are always selecting, right. There is a strong selection pressure to kill the organisms that are weaker, right. So, it is like you just take your mosquito spray and you spray it, you are trying to kill mosquitoes, at the same time you are selecting for resistant mosquitoes, right, and when you put two sprays you will find mosquitoes that are resistant to both sprays and so on, right. here the idea is but the flip side of it is that if you have two weak hits, right. So, R1 or G1, let us stick to gene for the moment. If you remove G1, nothing happens. So, there is no major selection pressure arising out of G1. You remove G2, nothing happens, no major selection pressure. You remove G1 and G2, the organism dies, right. So, these are two weak hits that can essentially trick the organism. So, there is some interesting studies about how weak hits can confuse complex systems. So, the idea is you give two hits that are not very strong, right. You are not targeting essential genes, but you are targeting important genes which the organism does not truly require for survival except together, right. An even better case for the application of synthetic lethals is in cancer therapy, right. So, cancer invariably involves some gene expression gone awry, some genes gone awry, right. So, what you find is you may have a cancer genome wherein a cell wherein it is already missing a particular gene. The loss of that potential tumor suppressor gene has induced the development of cancer in this cell, right. So, synthetic lethal is not singly lethal. So, the thing is, let us say so this is a healthy cell and this is a cancer cell. The cancer cell already lacks G1, right. The healthy cell has G1, right. Now, if you target G2, which is synthetically lethal with this, right, in both, right, the healthy cell will continue to live, the cancer cell will die. Is that clear? Because the cancer cell has already lost G1 in the process of developing cancer, maybe that is the reason it developed cancer or whatever, right. So, this is a very interesting aspect of synthetic lethals, not that you can use metabolic networks to find synthetic lethals in cancer, but I think that should be possible once we have better curated models and potentially better objective functions to study larger organisms, because the objective function challenge still very much remains, right. How do you actually find the most correct objective function for a complex cell? For a bacterial cell it is easy, maximize growth, cell is happy, we are happy because we get good predictability, but what about more complex organisms? Right, so, cancer therapy is another possible application for synthetic lethals. So, how do you identify synthetic lethals? What would be the simplest way to identify synthetic lethals, right. So, let us first look at experimental methods before we think about computational methods. So, yeast synthetic lethals have been identified using yeast synthetic genetic arrays. So, large high throughput phenotypic screens, you just remove multiple genes at a time and check the phenotype, right. So, previous in silico approaches also have built on the framework of FBA, but of course, it is restricted to metabolic genes always, right. So, all through the last few classes we have been restricting ourselves to metabolic genes. You cannot say what happens when you remove a non-metabolic gene. So, how would you identify synthetic lethals? What would be the naive way to identify synthetic lethals?
What have we found was the naive way, right? Removing one gene.
So, you just remove all pairs of genes, right. So, you have n genes in an organism, you make n choose 2 deletions and you figure out you can run an FBA for each of those cases or even a MOMA or room and then figure out whether you are getting a growth or not, right. So, single lethals are actually even easier to identify, it is just solving one optimization per deletion. Synthetic lethals, it is there is a combinatorial explosion. If you have 1000 genes, you are talking about 1000 choose 2, which is half a million. The problem is 1000 choose 3, which could be as high as quarter of a like 1/5 of a billion. So, this quickly becomes infeasible for larger organisms, but given the simulations are independent, it is easy to parallelize these across a cluster. So, this is commonly done, you just offload all this heavy computation to multiple cores which can do the job in parallel, because none of these simulations are dependent on one another. It is about setting V1 V2 equal to 0 in one simulation, V1 V3 equals 0 in another, V1 V4 equals 0 in another simulation. So, these are the constraints that you are throwing at literally the same core linear programming problem. The core LP is the same, maximize C transpose V such that SV equal to 0, subject to varying additional constraints on what is being deleted and so on.
So, but C will be same, right?
C is the same. So, there are previous approaches that have used bilevel mixed integer linear programming problems. So, there is this approach called SL finder, which poses the synthetic lethal identification problem very elegantly as a bilevel mixed integer linear programming problem. So, they identified synthetic lethal double and triple reaction deletions in E. coli, but the problem with MILP is that they become incrementally difficult to solve. So, how this works is I will not get into the formulation details, you can look it up. It first gets one synthetic lethal, right. It tries to solve an integer problem by saying delete at most 2 genes to give me a lethal phenotype, right. But while doing that, to get the next lethal you have to do what is known as an integer cut, something the equivalent of saying give me something other than the first solution. The next time you have to tell me give me something other than the first or the second solution, and so on. So, the problem becomes incrementally difficult to solve, and they took about 1 week to solve this on a workstation. But then a newer approach was developed called MCS enumerator. This is another MILP based method which runs much faster, right. But the issue with this in general is that you cannot parallelize these, because you want to use one solution to then say do not give me that solution and give me a better solution kind of thing, right. So, the question we wanted to ask was, is there a way to surmount the complexity of exhaustive enumeration and bilevel MILP, right. Exhaustive enumeration is obviously a lot of work. It is very difficult to do 170 million simulations if you are trying for triple lethals. Similarly, it is pretty difficult to do bilevel MILP problem solving as well. So, is there a way to get around this? I hope you got a good overview of the concept of synthetic lethals today and how different approaches have been used to identify synthetic lethals, but I also must have convinced you that these approaches are a little computationally expensive and we need alternative approaches. So, in the next video I will talk to you about a very interesting algorithm that was developed in our lab for computing synthetic lethals, namely the fast SL algorithm.